Our next speaker is Professor Tao Dong. So Professor Dong obtained her Doctor of Philosophy from Oxford University in the Nuffield Department of Medicine. She is now a professor of immunology at the university, as well as the program leader of the MRC Human Immunology Unit. She's also the founding director of both the CAMS Oxford Joint International Center for Translational Immunology and the Chinese Academy of Medical Sciences Oxford Institute. The main objective of Tao's group is the functional aspects of the antigen-specific T cells and studying the factors affecting T cells in controlling virus infection and cancer development. By understanding the key factors required for efficient viral control by the T cell responses in a number of different viral infections and for efficient viral control by the T cell responses, they aim to identify targets to augment and control the immune response as a way of improving the outcome in several important human diseases. So she's had several very exciting publications recently related to COVID-19, and she will be sharing with us her work on memory T cells primed by SARS-CoV-2 virus infection in UK COVID patients. All right.
Thank you, Emily, for your very kind introduction, and ProImmune for invitation, especially Jeremy, who is also our collaborators. Here, I would like to share our recent work on studying the SARS-CoV-2 specific T cells in COVID recovered patients in UK. So we know that cytotoxic T cells not only play an important role in protection against acute virus infection, but also play a central role in chronic virus infection, autoimmunity, and cancer. So my lab over the past many years has been focused on three key determinants, which we believe are most important in affecting the quality of human cytotoxic T cells, particularly influenza virus, HIV infection, and more recently, virus-associated cancer. So as we know, for COVID-19, the role of adaptive T cell immune responses in disease pathogenesis and the longer-term protective immunity is currently poorly defined, mainly because it's new virus, but it's essential to inform potential therapeutic intervention and enhance the vaccine design. So there are several ongoing vaccine trials, as we all know, and anxious about at the moment, but it is still not know yet and whether they will provide long-lasting protective immunity. Most vaccines are aimed to induce humoral immunity to the spike protein, but it is not yet known if this will be sufficient to induce full protective immunity to SARS-CoV-2. So studying the natural immunity to the virus, especially the role of SARS-CoV-2 specific T cells, therefore it's critical to fill the current knowledge gap for improved vaccine design. Since this is a new virus and we know very little about it, its natural immunity. So when we started to become very interested, and in early April, and we just want to ask some very basic questions. For example, what part of the virus T cell are targeting? Then what the immune memory like in patients recovered from COVID-19? And is there any association with disease outcome? So to be able to answer those questions, we recruited 42 individuals following recovery from COVID-19 in Oxford, including 28 mild cases and 14 severe cases. In addition, we also have 16 control individuals sampled before COVID-19 season, and so in parallel. So, and we didn't see any significant differences in gender or age between the mild and the severe groups, as you can see here. So we used well-established, quite very conventional method in our lab and two-dimensional ELISpot assays to start with, and with made 423 overlapping peptides spanning the whole SARS-CoV-2 proteasome, apart from ORF1, the reason we excluded from ORF1 is because it's so big, and also from the Journal Immunology paper published by a colleague of us, Xiaoning Xu and Chris Lee, and ORF1 seems like not very immunodominant. So Chris suggested us to exclude this protein, which is, I think, is a quite nice suggestion. And so this figure shows the overall distribution and the magnitude and the breadth of the interferon gamma responses against all virus protein peptide pools we tested. And the figure on the left showed the magnitude of interferon gamma T cell responses from each individual. Each bar shows the total T cell responses of each individual's responses. And each colored segment represents the source protein corresponding to the peptide pools, which eliciting interferon gamma T cell responses. And the figure on the right shows the breadth of the T cell responses from each individual. And the breadth of T cell responses was calculated by the number of peptide pools in the first dimensions. As you can see, the majority of the participants exhibited SARS-CoV-2 memory T cell responses to at least one of the peptides. So the breadth and the magnitude of T cell responses varied considerably between the individuals. T cell responses were detected against the peptide epitopes distributed across a wide variety of the virus protein, as you can see here, and a significant higher magnitude and broader breadth. Overall T cell responses were observed in the severe cases in comparison with mild cases. And in particular, responses to spike, membrane OIF3 and OIF8 proteins. However, we did not observe ex vivo interferon gamma producing SARS-CoV-2 specific T cell responses in healthy volunteers who were all sampled before any chance of exposure. But in those with appropriate HLA types, T cell responses were observed in the influenza virus, EBV, CMV using pools of known T cell epitopes, as well as PHA as positive controls. I think this is a kind of something probably unexposed individual is worth lots of study. I think the reason we didn't see the response as what other people see is probably because we use relatively low peptide concentration. Also, the number of input cells is also low. But I think it's definitely, and because we only studied 16 examples and in UK, so I think there are lots of questions and very important questions need to be addressed in this particular healthy individuals and the type of response and level of cross-reactivity and antigen specificity versus sensitivity. So it's a lot of I think the study need to be in-depth study need to be done, but I do believe there's definitely a level of cross-reactivity which are important, probably play a role here. So next we assessed a relationship between the spike specific and overall T cell responses in association with spike RBD and NP, specific antibody endpoint titers. And we found a significant correlation between the antibody titers and the T cell responses. And moreover, we see a significantly higher level of spike, RBD, and NP antibodies were observed in severe cases in comparison with mild cases. So this experiment was done by Nui and the team in Jules and Agavin's lab. So having identified overall T cell responses to SARS-CoV-2 peptide, and the responses detected against the positive peptide pools were characterized further for involvement of CD4 and CD8 T cell subsets in peptide recognition by evaluating specific intracellular production of interferon gamma, TNF-alpha, and IL-2 by T cell upon stimulation. So these slides are some examples of multi-cytokine analysis after stimulation of positive peptide pools using PBMCs isolated from individuals recovered from the mild and severe illness. So we found a greater proportion of T cell responses to spike and MNMP to be mediated by CD8 rather than CD4 T cells in those with mild disease compared to those with severe disease. We then performed multi-cytokine analysis, which revealed a similar pattern of interferon gamma, TNF-alpha, and IL-2 production by CD4 and CD8 T cells in both mild and severe cases. Interestingly, the multifunctional MNMP specific CD8 T cells were significantly higher than spike specific T cells in those with mild disease, but not in those with the severe disease. We also observed very low CD107 positive CD4 T cells overall, which suggests cytotoxic CD4 T cells might not be a major contributor to the virus clearance. And the interferon ELISpot assay we performed with candidate peptide identified from a two-dimensional matrix analysis in 34 subjects, and a total of 41 peptides containing T cell epitope region were recognized by COVID-19 recovered subject, 17 from spike, 10 from MP, and 6 from membrane, and 7 from ORF proteins. Strikingly, we found 6 dominant peptides were dominant peptide region were recognized by more than 18% of subjects tested, and one of them is 47% of subjects responded to the peptide M24, which is very striking, and we never see something like this in other virus infection. And those dominant responses were further confirmed by both ex vivo and using cultured short-term T cell lines, as example showed on the right panel of these studies. And so at the HLA restriction and the selected responses were further confirmed by identification of shared HLA typing between responding donors and the in vitro HLA restriction assay. So this is an example slides showing how we defined optimal epitopes and the HLA restriction use a panel of HLA matched EBV transformed B cell lines as a target cell for T cell recognition and the HLA restriction. So the optimal epitopes recognized with the lung peptides to which dominant CD8 response were detected using optimal epitope prediction is run by IEDB analysis tool. In total, 6 CD8 positive T cell epitopes restricted by HLA A0101, A301, and A11, B7, B4001, and B2705 were confirmed, as shown in the table on the left. Actually, peptide pentamer was synthesized by ProImmune, comprising 5 peptides bound to the appropriate HLA class 1 molecules we defined. And the right panel shows the example of pentamer staining. And strikingly, and despite almost half of the individuals possess HLA-A2, we were not able to identify any dominant A2 epitopes. A pentamer folded with HLA-A2 with the spike epitope reported previously by other groups was also synthesized by the ProImmune, and we only see 1 out of 6 A2 positive donors tested, and which shows very weak responses and with very low frequency. So the ex vivo tetramer staining also showed typical central memory and effector memory and early and or intermediate differentiation phenotype of SARS-CoV-2 specific T cells in recovered patients. Because the number of patients we studied is not very big, so we didn't really do a statistic analysis in comparing mild and severe, which we are doing at the moment. hopefully we will have some clear answer soon. So in summary, we found robust memory T cell responses specific for SARS-CoV-2 in the blood of donors who have recovered from COVID-19. The breadth, magnitude, and frequency of the memory T cell responses was significantly higher in severe compared to mild COVID-19 cases. The T cell responses correlated with antibody towards the spike and RBD, and a particular high frequency of spike protein specific CD4 T cell responses were observed in patients who had recovered from COVID-19. And furthermore, we identified epitope clusters and targeted by T cell across the donors, and a high proportion of SARS-CoV-2 specific CD4 to CD8 to CD4 T cell responses is also proposed in the mild cases. As always, there are still a lot more to do, and we have more questions, I'm afraid, than answers. And our group is now focusing on those following up this subject we studied and on the dominant responses identified, and we try to understand in detail what might went wrong with the T cells and how they might lost control or even contributed to severe illness in those very sick COVID patients. We're also generating more tools in order to address those questions properly. For example, a system we could evaluate how those T cells react to the virus-infected cells instead using peptide post target cells. So this is probably the most important slides, and what I presented today is really a great teamwork involving lots of people in Oxford and other universities in UK. The list is probably too long, and I apologize, many names are not listed here. The experimental work and the T cell assay are mainly done by people in my group, Yan Chunpeng, Gui Hailiu, Xuan Yao, Zi Xiying, and Dan Yingdong in MRC Human Immunology Unit, headed by Graham Oak. And antibody data were provided, generated by Nui in Zhu and Gavin's group. And we're really grateful for ProImmune for their great advice in optimal epitope prediction and their support in supply of what best quality pentamers, as you all agree, and the critical patient cohort and clinical information and relevant data analysis were provided by Alex Mensa and Julie Knight at the Wellcome Trust Center for Genetics. And all members, and I also like to acknowledge all members of the 4C Consortium, which is a important consortium and effort from lots of people to ensure the protocol and approval in place for the national-wide COVID immunology to be done, work to be done without any delay, especially at the early days. Also, Oxford Immunology Network COVID-19 response T-cell consortium for the stimulating discussion during our weekly meetings. And finally, patients and their families who gave the consent and participated study, and the funding from Medical Research Council to MRC Human Immunology Unit, and the CAMS Innovation Fund to CAMS Oxford Institute, which both based in the Oxford University. So I stop here and take questions. Thank you.
Great, thank you very much, Tao, for that data and taking us through the story of all that analysis. So we've got some time for questions, which is great. We do have a question, first of all, from Jason. Jason, if you are able to put your microphone on, then I will unmute you.
Okay, thanks, Dr. Dong, for your very impressive talk. I'm just curious about for your data talking about immunity against spike protein. Did you try to differentiate any immunity specifically on the S2 plant domain, which is the critical functional domain related with the fusion?
Yes, this is a very good question. Yes, we did, and we looked very carefully. But unfortunately, all the peptides so far we defined against spike is not in the RBD, and there are a lot in the C-terminal end of quite a few in the C-terminal end of the spike, but not in the RBD region, which is quite different from flu, I think.
Thank you very much, Jason, for your question. Our next question comes from Antonio Bertoletti. I'll unmute you, Antonio. You can ask your question.
Hi, Antonio.
Hi,
Yes,
Tao.
yes. Hi.
Very nice work, like usually. I have a question which is related about the difference that you find in severe cases and mild cases.
Yeah.
And my question is mainly due to the fact that we see, I would say, a sort of, let's say, decay of the T cell frequency after, let's say, clearance. I mean, did you know what were, let's say the patients that you study were matching in terms of time after, let's say, clearance?
Yes. So yeah. Okay.
Yeah. No. Please.
Yeah. So the patient we studied is all between 40 to 60 days after onset of symptom. So we did a correlation analysis. We did. I think it's like as all the T cell response, they all kind of decline. But I think between the mild and severe cases, when we compare the age and the time, there's no big difference. But I think it's as we kind of discussed, and it's a cause and the effect, I guess. It really depends, because you can imagine the severe cases, you have more virus and a longer exposure, so you would end up with more T cell response. So I'm actually not surprised at all. What we see is all expected, but I think the timing and the specificity is very important. And also because for this virus, it's so many things that's quite unusual, I think. And this actually, and the immunodominance, and also the protein specific. And so there's lots of things I think is interesting. And I think what we observed here as a kind of bulk analysis is more of the effect of the high and the longer viral exposure. And so we see this overall. So we have to dissect, probably even down to the individual response. And for example, like what you group are very good at, the T cell receptor, the clonality, even down to the single epitope specificity. I think is a lot need to be done in detail probably to get answer. And it's the data we provided here is more like a general what happened to COVID and what's left in the memory, I guess. Yeah,
we have a lot of things to do. Thank you. Very nice work anyway.
Exactly.
To everybody.
Great. Thank you, Antonia. Thank you for joining us quite so late in Singapore as well. We really appreciate it. Tao, I have a question, just thinking about next steps.
Yeah.
For you, what do you think, well, maybe two parts of the question. What have been the biggest challenges in getting the data that you've managed to get and published in time? What were the biggest challenges for you in that, maybe from a practical perspective? And then second, what are the, for you, what's the most exciting next step for the work?
Well, this is all not very, I think it's it's what's the most challenging thing. I think it's is at the beginning, the planning, I think. The planning and try not to go straight to the questions is likely, not complications, how to simplify the project as much as we can and go straight to the key questions we want. I think for this, it's more for us, we need to generate in the tools rather than to get answer. So because this is a brand new virus, we don't know anything about epitopes, everything. So it takes us, but there's so many questions you can answer, like ask for the T cell. So I think it takes us some time to decide what are the key experiments we want to do. And so that it was quite, I wouldn't say challenging, but I think that was quite critical for us, I think, at that time. And for the second question, I think the most exciting things for us, we really want to follow up this to understand why there is a cluster, is the immunodominance, and also the why there's a different kind of actually distribution. I feel like there's got to be a reason for why A2 don't, because you have A2 is a kind of standard. And Tony is shaking his head. Probably you find more A2. I don't know. I think it's maybe it's just unlucky. We just don't see much, but we see such strong like B7, B40. So everything else is so dominant. If you have that, actually half of the patient will respond. Don't know whether. But with A2, we have 50%. We do see but not dominant. I mean, the epitope. You do see the response, but why?
Yeah. Now, if I can add a small point, I mean, I agree with you that we also start to find more HLA B and C restricted T cells. But there is an interesting work on bioarchive that I saw that where they found A2 restricted response very high, but mainly on a ORF1 AB genome.
Yeah.
So maybe that's a different.
Maybe it's different. Yeah, maybe it's different. It's we don't know, but I'm just struck to find there is yeah. So
Great.
there's a lot, and we're establishing the virus infection system, which I think we are very excited. So that probably will tell us more about how T cell responds to the real virus infected cells. So I think that's also quite important.
Yeah.
Perfect.
Great.
Well, thank you again, Tom, very much indeed. We really appreciate your talk.